everyone, this is Nagavi, Assistant Professor from Department of Computer Science. Today we are going to say about the concept elements of visual perception in digital image processing. The elements of visual perception includes the structure of human eye, how the image is formed in the human eye, the brightness, adaptation and discrimination. First let us say the structure of human eye. The human eye includes the cornea and sclera as an outer membrane. The choroid element contains the blood vessels which is necessary for eye nutrition, and this element is pigmented in order to reduce the light entrance. Next, the choroid also includes the ciliary body, iris, diaphragm and the lens. The choroid controls the amount of light that enters the pupil. The lens is made up of fibrous cells which is suspended by the fibers. It is attached to the ciliary body. The lens is yellow in color. It absorbs 8% of visible light spectrum. It contains 60% of water and 6% of fat. The retina lies in the posterior position of the eye. Light receptors are placed over the surface of the retina. The retina includes two types of receptors. One is rods and another one is cones. The cones are also called as photopic or bright light vision. Rods are called as a scotopic or dim light vision. The horns are centered at the fovea, and these cones are highly sensitive to color. Cones are generally include red, green and blue, and the rods are useful for a general view. Next let us see mesophic vision. This is an intermediate region that helps us to differentiate between the bright and the dim light. Next, fovea. The structure of fovea is circular. It is 1.5 millimeter in diameter. The density of cones for fovea is 1,50,000 elements to 3,37,000. Next, blind spot. A blind spot is the region that emerges from the optic nerve of an eye. It is a place where the optic nerve connects to the retina, and it does not include any light sensors. Next, let us say the image formation in the eye. The eye lens is flexible when compared to the optical lens. It controls the tension in the fibers of the ciliary body. It also helps us to focus the distant objects and to focus the objects that are nearby. It helps us to focus the nearsighted and far-sighted objects. The distance between the center of lens and the retina varies from 17 mm to 14 mm. Perception takes place by the light receptors. The receptors transform the energy that are decoded by the brain. Next, let us say few terms. Luminance or intensity. Luminance is nothing but the light that is reflected or emitted from an object. The luminance of an object is independent of the luminance of the surrounding object. Next comes brightness. A brightness is the perceived luminance. When two objects with the different surroundings will have a same luminance, but the brightness will be different. Next, the contrast. The contrast is the difference in the visual properties that helps us to distinguish an object from other objects. Brightness is the perceived luminance. Next, brightness adaptation. At a given time, our human eye can discriminate only a smaller range that is called as the brightness adaptation. The dynamic range that a human visual system perceives is from 10 to the power minus 6 to 10 to the power 4, but it cannot accomplish this range simultaneously. The current sensitivity of the visual system is called as the brightness adaptation level. In the image given below, the y-axis represents the brightness and the x-axis represents the log intensities. Intensity is increasing from left to right, that is minus 6 to minus 4. The adaptation range is limited because there is a glare limit. Beyond this, we cannot identify anything because in this range the intensity will be more. So the scotophic vision becomes zero at this range, and then the photophic we can able to visualize the colors. As in for the scotophic the intensity becomes zero, the photophic intensity starts to increase so that we are able to visualize different colors. Next comes brightness discrimination. This brightness discrimination is done using Weber ratio. In Weber ratio, I indicates the background illumination. The delta IC indicates the increment of illumination. The smaller Weber ratio indicates the good discrimination, and the larger Weber ratio indicates the poor discrimination. Next, psychovisual effects. Intensity is not a function of a perceived brightness. It also includes three other parameters such as match band effect, simultaneous contrast, and optical illusion. First, let us see match band effect. In this, the intensities of a surrounding points will affect the perceived brightness at each point. As we see the image given below, the intensities appears darker on the left side, and it when comes to the right it becomes brighter. Next comes simultaneous contrast. All small squares in the image have exactly the same intensity, but only the background keeps changing because of a various contrast. This is called as a simultaneous contrast. Next comes optical illusion. Optical illusions are also known as the visual illusions. These are the misinterpretation caused by brain. When coming to image processing, we will program using a Python or a MATLAB. Let us see what is a MATLAB. MATLAB is a program for doing numerical computation. It was first designed for solving linear algebra type problems using matrices. The MATLAB is derived from a name Matrix Laboratory. MATLAB also has some built-in functions for solving problems such as data analysis, signal processing, optimization, and several other types of scientific computations. It also includes graphics and animations for both 2D and 3D. When you enter into a MATLAB environment, you will be able to see the command window where you will give all the MATLAB commands. The workspace shows all the current working variables. The history shows all the commands that you have used in the command window. Apart from using the commands, you can also write your code in the MATLAB using the editor menu. All the MATLAB files are open using this editor. All the MATLAB files are stored with the extension datem. Next, let us see some of the basic commands. What command list all the M files in the current directory. Dir list all M files in the current directory. Type test displays the test MATLAB file in the command window. Delete test deletes the test MATLAB file. Chdir helps to change the directory. Pwt shows the current working directory. Who lists the known variables? Who lists known variables with a size? Clear Clear and CLC both clear variables from the workspace. MATLAB and matrices. MATLAB treats all the variables as a matrices. Vectors are also a special forms of a matrices, but the difference between the matrices and the vectors are matrices are two-dimensional, whereas the vectors are one-dimensional. Next, the variable names. Variable names in MATLAB are case sensitive. The variable names can contain up to 63 characters. The variable names must start with the letter followed by letters, digits, and underscores. MATLAB variables are defined by assignment. There is no need to declare in the advance the variables that we want to use in our program. These are some of the examples for variable declarations. Every statement must end with a semicolon in order to suppress the output. There are some other symbols in the MATLAB such as double greater than symbol indicates the prompt. The three dots indicates the statement on the next line. The comma is used to separate the statements and the data. The percentage symbol is used to represent the start command. Semicolon is used to suppress the output. It also used in matrices for separating the rows and columns. Colon symbol is used to specify a range. Some matrix functions that is used to create in a MATLAB. The zeros of rows, comma columns. This function will create a zero matrix. Rand of rows, comma columns generate a random matrix. Ones of rows, comma columns create a matrix with all the entries as 1. I of rows, comma columns create an identity matrix. We can also extract a submatrix from a matrix. Let us see some examples in the MATLAB.